everyone. My name is Fernanda Martinez, and I'll be discussing a crucial topic in vector control, which is the biological control of mosquitoes. This presentation is based on Chapter 43 of Biology of Disease Vectors, and I will talk about how natural and applied biological control methods help in reducing mosquito populations. Hopefully, by the end, you'll understand different control strategies, their effectiveness, and challenges in implementing them.
So this is just a quick overview of what I will talk about in this presentation and the key ideas or key terms that are discussed in this chapter. So I will introduce the types of biological control, the key principles, the challenges in implementing biological control, and the future directions that will head into biological control. So, did you know that 70% of all infectious diseases in humans originate from animals? These are called zoonotic diseases, and of these, 20% are transmitted by vectors like mosquitoes. Mosquitoes are among the most significant vectors responsible for spreading deadly diseases such as malaria, dengue, and the Zika virus. This makes controlling their populations a critical priority for global public health. Traditionally, insecticides have been the go-to solution for mosquito control, both in households and commercial settings. However, their widespread use has led to two major challenges. First, mosquitoes are increasingly developing resistance to these chemicals, rendering them less effective over time. And second, the overuse of insecticides poses serious environmental risks, harming ecosystems and non-target species. In light of these challenges, biological control has emerged as a more sustainable and effective alternative. So, what is biological control? Biological control refers to the deliberate reduction of pest populations through the introduction of natural predators, pathogens, parasites, competitors, or microbial toxins. This strategy was first documented in 1889 when a predatory insect was introduced to combat a scale insect infestation threatening California's citrus industry. Over time, biological control expanded significantly, particularly in agriculture, as an environmentally sustainable alternative to chemical pesticides. In the early 20th century, biological control gained widespread attention with the introduction of the mosquito fish as a means to regulate mosquito populations. However, this approach was largely abandoned in the 1940s and 50s following the discovery and widespread application of DDT. By the 1960s, growing concerns over the environmental and ecological consequences of large-scale insecticide use reignited interest in biological control as a safer and more sustainable alternative. One of the primary advantages of biological control lies in its target specificity, minimizing unintended harm to non-target organisms. Additionally, it offers minimal environmental impact compared to chemical insecticides, which can lead to their resistance and ecological imbalance. Furthermore, some biological control agents provide long-term regulation after a single introduction, making them a cost-effective solution in integrated vector management programs. And this image here illustrates the mosquito fish, which remains the key biological control agent in aquatic environments. So in this slide, I will discuss density-dependent and density-independent mortality factors, which are critical in shaping mosquito population dynamics, as illustrated in figure 43.1. These factors regulate population sizes through different mechanisms, influencing vector control strategies. So, density-dependent factors increase in severity as population density rises and decrease when populations are smaller. These factors naturally regulate populations by keeping them around an average size, as shown in figure 43.1a, where the mosquito population fluctuates around K. K represents the theoretical carrying capacity or the maximum population size that the environment can sustain given available resources such as food, space, and breeding sites. Once the population exceeds K, interspecific competition increases, leading to the resource depletion and a decline in population growth. Essentially, density-dependent factors act as a natural brake on population growth. If the numbers rise too high, factors like pathogens, predators, or competition reduce the population. Conversely, if the population shrinks, then density-dependent factors ease off, allowing the numbers to rebound. However, in reality, mosquito populations do not always stabilize around K. This is represented by figure 43.1b. An example where a density-dependent factor such as a predator or a pathogen maintains a mosquito population at an average size. However, in some cases, killing excess mosquitoes may not effectively reduce their numbers due to overcompensation. With fewer larvae competing for resources, more survive to adulthood, counteracting the control efforts. For diseases like malaria, the primary goal is to reduce the number of adult mosquitoes, as only adult females transmit the disease. Therefore, larval biological control agents are not widely used in malaria control, since reducing larvae does not necessarily translate to a decrease in adult vectors. Instead, control efforts focus on targeting adult mosquitoes through insecticides, genetic modification, or biological control agents that directly impact adult survival. On the other hand, density-independent mortality factors cause fluctuations in population size regardless of density. These include environmental factors such as floods, droughts, extreme temperatures, and insecticides, which can lead to significant population crashes. Figure 43.1c illustrates the impact of a single density-independent event, where 80% of the mosquito population is eliminated. Over time, in the absence of further DI events, the population recovers to its carrying capacity. Figure 43.1d further emphasizes how recurrent DI events can drastically alter population dynamics. So understanding these ecological principles is fundamental in designing effective vector control strategies. While density-dependent factors maintain population equilibrium, they may not always lead to long-term suppression of disease vectors. In contrast, density-independent factors such as insecticide application or climate events can cause dramatic population declines, but may not offer sustained control unless strategically applied in conjunction with other methods. While mosquito species exhibit significant ecological variation, certain patterns can be generalized and observed across populations. Mosquitoes are often characterized by having rapid population expansions and sudden declines, driven by their high fecundity and short generation times, particularly in tropical regions. This means that populations can increase explosively at the onset of the breeding season or rebound swiftly following catastrophic events such as droughts or insecticide applications. Additionally, mosquito species display habitat plasticity, shifting their preferred breeding sites in response to seasonal changes and environmental availability. Larvae are not evenly distributed across all potential breeding sites. Instead, they tend to aggregate in larger, more stable habitats and may colonize only a fraction of smaller sites, such as artificial containers. The diversity of larval habitats range from temporary puddles to permanent water bodies, and from artificial to fully natural environments. And this complicates efforts to implement a single, universally effective biological control strategy. So, how do these ecological traits influence biological control efforts? The ideal mosquito biological control agent, in theory, would need to possess adaptive traits that mirror those of its target vector species. It would have to respond swiftly to population surges, rapidly colonize diverse and shifting habitats, and produce offspring in numbers sufficient to counteract mosquito reproduction. However, no single biological control agent currently meets these criteria comprehensively. This necessitates an integrated approach that combines multiple agents such as predators, pathogens, and environmental modification to achieve sustained and effective vector suppression. Biological control can be broadly categorized into two main approaches: natural and applied. Natural biological control refers to the suppression of vector populations by naturally occurring predators, pathogens, and competitors. This type of control is most effective in stable, permanent habitats as opposed to temporary or fluctuating environments. While natural biological control plays a crucial role in regulating mosquito populations, it is rarely sufficient on its own to halt disease transmission. Interestingly, when natural control mechanisms are disrupted, whether due to environmental changes or the use of pesticides, we often observe a rapid surge in vector populations. This underscores the critical importance of these natural checks in maintaining ecological balance and controlling mosquito numbers. The second approach, applied biological control, involves deliberate human intervention to introduce, enhance, or protect biological control agents within a specific area. This could include timing the release of pathogens to maximize their impact on mosquito populations, or introduce large number of natural predators or parasites to bolster suppression efforts. Within applied biological control, we can further distinguish between two key strategies: augmentation and conservation. Augmentation entails the intentional release of natural enemies such as predators, parasites, or pathogens into vector habitats. The goal is for these introduced organisms to reproduce and establish themselves, thereby providing sustained control over multiple generations. This method is particularly valuable in regions where natural enemies exist but are not sufficiently abundant to exert a significant impact. Conversely, conservation focuses on optimizing existing natural biological control by safeguarding beneficial species and enhancing their efficacy. This might involve reducing pesticide use to protect mosquito predators, improving breeding conditions for natural enemies, or creating habitats to support these organisms throughout the year. Both augmentation and conservation strategies are integral components of a broader framework known as Integrated Pest Management, or IPM. IPM is a holistic approach that assesses all available pest control methods, biological, chemical, and environmental, and strategically integrates them to achieve effective control while minimizing ecological harm. Rather than relying on a single method, IPM aims to create a balanced, sustainable, and long-term solution for vector control, ensuring public health and environmental preservation go hand in hand. Now I will go ahead and talk about the types of biological control agents that are mentioned in this chapter. They mention pathogens, predators, and nematodes. Pathogens include bacteria, viruses, fungi, protists. Predators are fish, insects, and crustaceans, and nematodes under their own category of parasitic worms. Pathogens play a key role in mosquito control by targeting larvae specifically. One of the most effective microbial agents is Bacillus thuringiensis israelensis, also known as BTI, which produces toxins that disrupt the larval gut, leading to their death. Another promising agent is the fungus Lagenidium giganteum, which infects mosquito larvae through motile zoospores that penetrate and grow inside the host, eventually killing it. Lagenidium is valuable due to its long-term persistence in aquatic environments, though environmental factors like pollution and temperature changes can reduce its effectiveness. These microbial agents provide an eco-friendly alternative to insecticides, but careful management is needed to prevent resistance and maintain long-term control. In contrast, viruses have not been effective for mosquito control. They infect only a small portion of the population, require high doses for infection, and become less effective as larvae mature. Many viruses are transferally transmitted, meaning that they pass from adults to offspring without significantly impacting the current generation, making them ineffective for rapid vector suppression. Because of these limitations, research has focused on bacteria like BTI, fungal agents like Legionnairium, and genetic modification techniques that directly suppress mosquito populations, offering more reliable and scalable control strategies. Protists, particularly Microspora, are common mosquito pathogens but typically cause low mortality and don't persist well in the environment, making them ineffective for large-scale vector control. One exception is Ambliospora species, a microsporidian with a complex life cycle. In figure 43.3, it is shown that infected female mosquitoes pass spores to their eggs, which develop into larvae carrying the parasite. The cycle continues when larvae release spores into the water where they infect an intermediate host, usually a coppod. When a mosquito larva consumes an infected copod, the parasite returns to its sexual phase, completing the cycle and infecting future mosquito generations. This natural control mechanism reduces mosquito populations without harming non-target species, but its effectiveness depends on environmental conditions and the presence of intermediate hosts, making it unpredictable for large-scale control. Many invertebrates, such as predatory insects, hydra, and flatworms, act as natural mosquito predators. Effective predators are often found at low densities because they naturally regulate mosquito populations. Unlike pathogens, predators are highly mobile and less sensitive to water quality. However, they typically reproduce more slowly than their prey, which can allow mosquito populations to occasionally escape their control. One example is Toxorhynchides species, a predatory mosquito that has been used to target container breeding mosquitoes. Despite its effectiveness, its low fecundity and longer life cycle compared to its prey make it less than ideal for widespread control. Fish are among the most widely used mosquito predators, with smaller fish being particularly efficient at consuming larvae. Gambusia affinis, or mosquito fish, is specifically effective due to its ability to thrive in low oxygen environments, tolerate a wide temperature range between 13 to 35 degrees Celsius, and adapt to salt water. As a surface feeder, it is highly attracted to moving prey, making it most effective in clear vegetation-free waters. However, Gambusia can also outcompete or prey on native species, including endangered fish like the desert pupfish and commercially valuable species. To mitigate these ecological risks, native fish should be prioritized for mosquito control whenever possible. Although insect parasitic nematodes are not microorganisms, their small size, persistence, and role in biological control align them with microbial pathogens. Several myrmidid nematodes parasitize mosquito and black fly larvae, but only Rhomenomyrmis has been developed for vector control. Female lays eggs in the substrate of mosquito larval habitats. The pre-parasitic stage attaches to a mosquito larva using a stylet, penetrates the cuticle, and develops inside the host. Interestingly, higher parasite densities within a host lead to more male nematodes, which influence population dynamics. Due to its long life cycle and reliance on hosts, R. culcivorax is most effective as an inundative biological control agent. This requires regular, large-scale introductions to achieve sustained mosquito suppression. While biological control offers many advantages, it also comes with challenges. Environmental factors like temperature, water quality, and pollution can impact the survival and effectiveness of control agents. Resistance is another concern. Mosquitoes can develop resistance to bacterial toxins over time, reducing long-term efficacy. Lastly, mass production and distribution of biological agents requires significant investment, making large-scale implementation a challenge. Addressing these limitations is crucial for sustainable mosquito control. While BTI and natural predators continue to be effective, advancements in biotechnology and genetic modification are revolutionizing mosquito control. One such approach is the sterile insect technique, where male mosquitoes are sterilized using radiation and then released into the wild. These sterilize male mates with wild females, preventing reproduction and leading to population decline without the use of pesticides. Additionally, gene-drive technology is being explored to introduce genetic modifications that rapidly spread through mosquito populations, ensuring long-term suppression of vector species. These emerging technologies offer innovative, sustainable, and scalable solutions for mosquito control, potentially transforming the way we combat vector-borne diseases. So these are my references. They provide a deeper insight into the effectiveness and potential of biological control strategies and mosquito management. Thank you for your time.